Hello everyone, and welcome to today's live broadcast. Antigen-specific polyfunctional CD4 positive T cells are required for vaccine-mediated protection in tularemia, presented by Dr. Lydia Roberts. I'm Christina Jewell of Labroots, and I'll be your moderator for today's event. We're excited to bring you this educational web seminar presented by Labroots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. Today's webinar is sponsored by Miltenyi Biotech. The Miltenyi Biotech mission is to improve scientific understanding and medical progress by providing products and services that advance biomedical research and cellular therapy. Honoring this mission drives their commitment to support the translation of basic research into therapy in the areas of immunology, cancer, neuroscience, and stem cell biology. For more information, please go to www.miltenyibiotech.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have any trouble seeing or hearing this presentation, please click on the support button at the top right of the presentation window. Or you can use the Q&A button to let us know you're having a problem. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce our speaker, Dr. Lydia Roberts. Dr. Roberts received her undergraduate degrees in chemistry and biochemistry from Kansas State University in 2008. Followed by her PhD in microbiology and immunology in 2013 from the University of North Carolina at Chapel Hill. Currently, she is postdoctoral fellow in the laboratory of Dr. Catherine Bozio at Rocky Mountain Laboratories, NIAID, NIH. Please join me in welcoming Dr. Lydia Roberts. I will now turn the presentation over to her. Dr. Roberts?
Thank you so much, Christy, and thank you to everyone who is attending this webinar today. I'd first like to thank Miltenyi Biotech for inviting me to present some of my postdoctoral research today. And I'd also like to thank Labroots for hosting this webinar. It's a really great way to share science. So as Christy said, I'm a postdoc in Katie Bozio's lab at Rocky Mountain Labs, which is a satellite campus of NIAID. And at RML, we study a lot of really interesting pathogens. And the pathogen that our laboratory studies is Francisella tularensis. Francisella is a highly virulent intracellular pathogen and the causative agent of tularemia. Now, tularemia can occur via multiple routes of infection, the most common being contracting the bacteria through a cut in the skin, which results in glandular tularemia. Another common and very deadly form of this disease is pneumonic tularemia, which is caused by inhalation of contaminated aerosols. Now, in the United States each year, there aren't very many naturally occurring cases of tularemia. The latest data from the CDC shows that there were 180 cases in the United States in 2014. And you can see that many of these cases are centered around the states of Oklahoma, Kansas, Missouri, and Arkansas. And so why are we interested in such a pathogen that doesn't really cause a tremendous public health burden by naturally occurring cases in the U.S.? Well, we're particularly interested in Francisella because it can be used and has been used as a bioweapon. Francisella was the infectious agent in bioweapons developed by both the United States and the former Soviet Union. And it's important to note that in the Soviet Union, these strains were engineered to be antibiotic resistant, thereby eliminating a very effective treatment for tularemia, which is antibiotic therapy. In the United States, Francisella is categorized as a tier 1 select agent because it has a very low infectious dose. As I indicated earlier, pneumonic tularemia is the most deadly form of this disease. As few as 10 inhaled organisms can be fatal up to 60% of the time if a person does not receive any antibiotic intervention. Francisella can also be easily aerosolized, and of course this allows the pathogen to many people to be exposed to this pathogen, again by the most virulent route. As I said before, Francisella has very high rates of morbidity and mortality, and it's an interesting bacterial pathogen in that it can infect nearly everything. It can infect small mammals, it can infect arthropod vectors such as ticks, it can even infect soil amoeba. So in the case of a bioterrorism attack, this pathogen will persist in the environment, which could allow for humans to then be a dead-end host for the infection. But it is important to note that Francisella is not transmitted person to person. It is a zoonotic pathogen. Now, for such a high consequence pathogen, ideally we would have a vaccine to protect not only the public but also our military from such a bioterrorism threat. And indeed, efforts were made in the 1950s to create a live vaccine strain, or LVS strain. And this was done by serially passaging the less virulent subspecies of Francisella tularensis, subspecies Holarctica. Holarctica was passaged on agar plates, it was passaged through animals, and it was an LVS strain that was attenuated was in fact generated. However, this vaccine is not licensed for use in the United States by the FDA because its mechanisms of attenuation are undefined. And furthermore, even if we did know the ways by which this strain of Francisella was attenuated, it isn't a very good vaccine. And this was known as early as the 1960s. In this classic paper here from McCrum, and I've modified a little bit to show the actual doses of SHU4 that the volunteers were challenged with. So I think this really well illustrates the ineffective nature of the current LVS vaccine. So in these experiments, individuals were either vaccinated with the LVS strain, which you can see here in this column, and then or they were left naive, which would be shown on the right. They were then challenged with increasing doses of the virulent strain of Francisella called SHU-S4, and you can see that, as I said before, with just 10 CFUs, the control patients all required antibiotic intervention, whereas the vaccinated individuals, only one had very mild symptoms. If you increase the dose to just 100 CFU, you can see that 3 of the people were fine, 1 had mild symptoms, and 1 had what was considered moderate symptoms. But at just 1,000 CFU, which is not a very high dose, particularly given this aerosol challenge, you can see here that 1 of these patients required antibiotic intervention, and the other 2 were not feeling too great and had what was considered moderate symptoms. In addition to Francisella, the LVS strain not protecting against very high doses of Francisella, we also know from the literature that protection wanes with time. And essentially there's no protective immunity approximately 1 year after vaccination with LVS. And so while there is a vaccine that exists, there's a lot of room for improvement for the LVS vaccine. And so this brings me to sort of the big questions that my project is focused on addressing. Most importantly, we're interested in understanding what components of the immune response mediate protection. If we can understand how a vaccine protects against tularemia, we can then use these identified components to make sure that rationally designed subunit vaccines elicit this protective response. And sort of in concert with this idea of understanding what components of the immune response mediate protection, we also were interested in understanding why the LVS strain that is currently out there does not protect against higher challenge doses and why protection wanes over time. And once again, if we can understand why these two things occur, we can more rationally design vaccines to eliminate these problems so that we have a very highly efficacious vaccine. So in order to answer these questions, we need to have an appropriate animal model. And I was very fortunate when I joined the lab in 2014, the senior postdoc in the laboratory was establishing this model in the black 6 mouse. And of course, it's important for a lot of immunologists to be able to use B6 mice because there are numerous immunological and genetic tools that are available only on this background. Also importantly, we were able to compare 2 different strains of LVS, ATCC and RML. The ATCC strain of LVS is the strain that's deposited in the American Tissue Culture Collection, which, to keep things simple, we've called ATCC LVS. This strain has been highly passaged, and unfortunately the passage history is sort of unknown. And so, but this is a strain that is commonly used by labs in the United States. We also had at our disposal a strain of LVS, which we've not very originally termed RML LVS, because it was a strain of LVS that was in a freezer that was a gift from Fran Nano. And its history is that it's one of the original vaccine vials when LVS was first generated back in the 1950s. And so we were going to started out by comparing these 2 strains of LVS. So the basic model that we use is all intranasal infections in C57 black 6 female mice. We challenged the mice with increasing doses of either ATCC or RML LVS and then waited 28 days. All of the mice that survived their primary infection were then intranasally challenged with virulent Francisella. It's called SHUS4. And we do, our challenge dose is 25 CFU, and this is a dose that is 100% lethal in naive animals. And then after this challenge, we measure survival. So as you can see here on the left, we have very different degrees of survival with our 2 LVS strains. So let's first focus on ATCC LVS, which is on the left. You can see that irrespective of the dose that the mice were vaccinated with, again, these are mice that survived this vaccination dose. Again, irrespective of the dose that they were vaccinated with, nearly all of the mice succumb to SHUS4 challenge, with only about 10% surviving if they were vaccinated with 1,000 CFU of ATCC. Now this is in very strong contrast to what we find when we look on the right at RML LVS. You can see here that we have only 2 groups of mice. We have mice that survived 100 CFU of RML LVS and the mice that survived 10 CFU. And so the first thing that this tells us is that there are very different LD50s of these strains. So the LD50 that we calculated for RML LVS is about 170 CFU, where for ATCC, the LD50 is almost 10,000. So clearly there are very different virulence characteristics of these 2 strains of LVS. Most importantly, however, is the ability of RML LVS to protect a B6 mouse when challenged with virulent Francisella. And so this was the first report, being able to protect a B6 mouse against virulent Francisella infection with a live vaccine strain of Francisella subsp holarctica. All right, so now that we have our mouse model, we wanted to take advantage of it and first look at what cells or cell types may be required for protection. So if you remember at the beginning, I told you that Francisella is an intracellular pathogen. And so it's not surprising that a prime candidate for a cell population that might be important would be T cells. T cells have been shown in the literature to be required for mice to survive a primary infection with LVS. And then another paper from Wayne Conlin's lab showed that BALB/C mice that had been vaccinated with LVS also required both CD4 and CD8 T cells to survive a virulent Francisella challenge. And so we wanted to test whether or not that was also true now that we're working in black 6 mice. So for these experiments, mice were vaccinated with RML LVS. Let's see here. And then 28 days later, they were challenged with virulent Francisella SHU-S4. Now before and then during the challenge period with SHU-4, mice were treated with either antibody to deplete CD4 positive T cells or CD8 positive T cells. And our results were particularly interesting because like the other groups, we did show that both CD4 and CD8 positive T cells are required for mice to survive, vaccinated mice to survive tularemia. However, we have this very important distinction in the timing of the requirement of these cells. So in red, which is right here, you can see that mice that lack CD4 positive T cells succumb very rapidly to virulent Francisella challenge. Whereas mice that were depleted of CD8 T cells, while they do succumb to infection, they do so significantly later on during the infection. And so what this indicated to us is that the mice require on-board, ready-to-go CD4 positive T cells in order to survive a primary or a secondary infection with virulent Francisella. And while CD8s are important, they probably play a role later on during infection. And so given this really important role and sort of temporal role for CD4 positive T cells, we really wanted to focus on this cell population for our initial studies. The little wrinkle that I sort of brought to the lab when I joined was that we really needed to be looking at Francisella-specific T cells. So up until this point, our lab had been doing quite a bit of adaptive immunity research, looking at the adaptive immune response to Francisella. However, we'd always been doing that sort of by looking at the entire T cell pool. So as you know, the only cells that are responding to infection are those that recognize antigens in Francisella. And so while we can gain a lot of important information by looking at the broad T cell response, if we can dissect out the response and specifically isolate Francisella-specific T cells and look at how those cells are responding to infection, we can learn a lot more about how those cells are mediating protection. So in order to do this, we need to have identified epitopes. And there is one immunodominant epitope that has been identified in the B6 mouse. It's in a lipoprotein called TOL4, and again, it has been shown to be immunodominant in black 6 mice. However, attempts to make MHC class II tetramer that recognize cells specific for this epitope has proven difficult. And so we needed to have another approach so that we could look at these antigen-specific cells. So what we decided to do instead was to generate recombinant strains. And these recombinant strains would express another well-characterized CD4 epitope that then would allow us to track those T cells using class II tetramer. Now the epitope that we chose to follow is from lymphocytic choriomeningitis virus, or LCMV. The immunodominant epitope is GP61 to 80. I'll further refer to this as GP61. We generated these strains by expressing them off a plasmid. We fused the GP61 epitope to the Francisella protein IGLC. IGLC is a highly expressed Francisella protein, particularly when Francisella is growing intracellularly. And this would allow a large amount of the protein to be expressed inside of cells. Francisella typically primarily targets dendritic cells and macrophages, and so it infects APCs. So there should be a large pool of antigen for those cells to present via the class 2 presentation pathway. And again, because there are the tools available to follow this epitope, we could use existing MHC class 2 tetramer to follow these T cells. And I just want to show here this Western blot. So again, we're expressing this epitope off of a plasmid. And so the bacteria have their endogenous level of IgLC, which you can see in the very thick band here on that row. And then what you should also be able to see are these smaller shifted bands here for each of our recombinant strains. So you can see that the shift is caused by the difference in size when we have this epitope expressed. And so these strains do have detectable protein. It's tagged with the GP61 epitope. Importantly, we also vaccinated mice with our RML and ATCC GP61 expressing strains. And then 28 days later, we challenged those mice with LCMV. And the only way that the mice would clear LCMV faster than the control vaccinated mice would be if there was a pool of GP61 specific T cells there to mediate that protection. And indeed, we did see a reduction in viral burdens in our mice that were vaccinated with the GP61 expressing strains compared to the overexpression of IgLC alone, indicating that indeed these strains are behaving as we expected them to, and the epitope is getting expressed. And so now that we have all of these really great tools, we can use B6 mice, we have these 2 different strains of LVS, ATCC, and RML that have differential protection. We have recombinant strains that can allow us to detect antigen-specific cells. We now can really test our hypothesis, which was whether or not there are quantitative and qualitative differences in the CD4 positive T cell response in mice that have been vaccinated with the protective strain, which again is RML, versus the non-protective strain, which is ATCC. So again, just so that everyone's clear about the model, we have B6 mice that are vaccinated intranasally with ATCC or RML expressing GP61. And then at various times post-infection, we are going to euthanize these animals and look for the presence of antigen-specific T cells by using flow cytometry. And then we also were interested in the function of these T cells, and so we also used intracellular cytokine staining to detect the presence of interferon gamma, TNF-alpha, and IL-2. So I'm going to start by showing data looking at the kinetics of the antigen-specific T cell response. So just to orient everyone, we're looking in 3 different organs. We're going to look in the lung, the mediastinal lymph node, which is the draining lymph node of the lung, as well as the spleen. And I also want to mention that Francisella or tularemia is a systemic disease. It will cause bacteria to not only be present in the lung after an intranasal infection, but you will also find bacteria in other organs such as the liver and in the spleen. And so here I'm showing data various days post-inoculation, again with either RML GP61, which is shown in the filled circles, or ATCC GP61, which is shown in open circles. And on the y-axis, we're going to count the total number of cells that stained with the GP61 tetramer that had an effector phenotype. So effector CD4 positive T cells are CD4 positive, CD44 positive, and CD62L negative. And so what you can see sort of right away is that in the lung, there is a nice antigen-specific T-cell response in ATCC vaccinated mice on day 10 post-infection. You can see that here. However, this response quickly wanes, and by day 28, there's essentially no detectable cells remaining in the lung from an ATCC vaccinated mouse. Now, interestingly, we never detected in the 2 independent experiments that I performed a large percentage or large pool of antigen-specific T cells in GP61 vaccinated mice. What we saw instead were a really nice pool in the mediastinal lymph node, as well as in the peripheral site of the spleen. And this occurred at day 14 post-vaccination. Now, what was most important about this experiment is the fact that RML GP61 vaccination resulted in a pool of T cells that were antigen-specific in both the mediastinal lymph node and in the spleen, 28 days post-vaccination. And this is important because 28 days post-vaccination is when we challenge mice with virulent Francisella US4, indicating that RML vaccination is eliciting more GP61-specific CD4 T cells, and that these cells are maintained into a memory phase and are present at the time of rechallenge. Okay, so now that we know that there are more antigen-specific cells after RML vaccination, we're really interested in the function of those cells. But what functions exactly do we want to look at, and what T-cell effector functions would be important for controlling a Francisella infection? So this experiment ended up being a really crucial experiment going forward for, I think, our entire laboratory, because it showed that in order to control virulent Francisella infection in our model, which is bone marrow-derived macrophages, in order to kill those bacteria, you must have both interferon gamma and TNF-alpha. Let me just walk you through these data. So on the y-axis, you'll see total CFU, and then on the x-axis, you'll see hours post-infection. And so the 3-hour time point gives you a sense of how many bacteria were taken up by the macrophages and are present at the time at which the cells were treated with either interferon gamma or TNF-alpha, or both of those cytokines. And what was really interesting about this result is that SHUS4 sort of laughs in the face at being treated with interferon gamma alone or TNF-alpha alone. It continues to replicate. It doesn't act as though anything is happening. It isn't until the cells, the bone marrow macrophages that are infected, have been treated with both interferon gamma and TNF-alpha that you actually begin to see a reduction in bacterial burdens, suggesting that the ability of a CD4 positive T cell to produce both interferon gamma and TNF-alpha will be most efficient and effective at controlling SHUS4 replication in a mouse. And this made us interested then in identifying the presence of polyfunctional CD4 positive T cells, so T cells that were making both interferon gamma and TNF-alpha. And importantly, we were also interested in whether or not those T cells were making IL-2, since IL-2 would further support the generation of T cells. And so in these data here, we enumerated the number of polyfunctional CD4 T cells in the lung and in the spleen. So let's first start here. We'll look in the lung. So what you can see in terms of what I call triple producers. So these are cells that are positive for interferon gamma, TNF-alpha, and IL-2. And you can see that in the lung, there are significantly more triple producers. They're polyfunctional in RML vaccinated mice compared to ATCC. And these data are all from day 28 post-vaccination, because this is the time point at which mice are challenged with virulent SHUS4. You can also see in the lung there are more double producers. So these are cells that are making interferon gamma and TNF-alpha, but not IL-2. Now in the spleen, when we look at triple producers, there wasn't a statistically significant increase in the number of triple producers in RML vaccinated mice compared to ATCC, but you can see it's trending that way. And then we also did see a significant increase in the number of double producers in the spleen in RML vaccinated mice compared to ATCC vaccinated mice. And so far I've shown you that we have more antigen-specific cells and that the cells that are present are, have greater effector function in that there are more polyfunctional CD4 positive T cells after RML vaccination compared to ATCC. Now I wanted to further evaluate the functional ability of these CD4 positive T cells after vaccination. And so to do this, we developed a culture system. And this system allows us in sort of a high throughput manner to evaluate whether or not a particular cell type is able to control SHU-S4 infection. So the experiments that I'm going to show you, we vaccinated mice again intranasally with ATCC or RML-GP61. We waited 28 days. The mice were then euthanized, and we purified CD4 positive T cells from both the spleen and the lung. And we overlay or co-culture these purified T cells onto bone marrow-derived macrophages that have been infected for 3 hours with virulent SHU-S4. We then wait 48 hours and then enumerate the number of CFUs that are present. And we can also look at secretion of cytokines like interferon gamma, but I'm only going to show CFU data to you today. Okay, so let's start on the left. Let's look at the lung. So again, I have total CFU on the y-axis, and then on the x-axis, we've got the number of CD4s that were overlaid, and this is times 10 to the 4. And so we've been overlaid 30,000 CD4 positive T cells, and in my wells, there are 96 wells, I've got 60,000 bone marrow-derived macrophages, so it's a 1-to-2 ratio of cells. Now I have different groups. So we've got a no overlay control. So this is bone marrow-derived macrophages alone. No cells are added to them. They're just sort of what would happen in that 48-hour time point, how many CFUs would be present. We also overlay purified CD4 positive T cells from naive animals as a control. And importantly, they look a lot like naive, no overlay. Naive mice and no overlay look the same, which is important and shows that our system is working as we expected. So what you can see when you compare, again, RML mice are in filled circles, ATCC are in open circles. You can see that immune mice do control the infection. Cells from the immune mice control infection significantly better than those from naive mice, which is what we would expect. However, there are no differences in the lung when you overlay purified CD4s from ATCC versus RML. However, if we look at the spleen here, you can see that when you start to titrate down the number of T cells that are added, there is a significant decrease in the number of CFUs recovered from RML vaccinated mice compared to when T cells are overlaid from ATCC vaccinated mice, suggesting that cells isolated from RML vaccinated mice are more functional than ATCC. Now this result was somewhat surprising given the fact that we didn't see differences in the number of polyfunctional cells when we looked at ICS. We didn't see differences in the spleen, not as great a differences that we saw in the lung. And so one of the avenues that we're pursuing now is that maybe it's possible when you take those cells out of the lung and then do them in this culture system, there's no architecture to separate the cells. So maybe if they're just sort of purified and put on, they're perfectly fine. But there's something about them in vivo that might prevent, they don't traffic properly, or they're not, they need to be resident in the airway and they're resident in the tissue. Those sorts of questions, we're trying to dissect that out, and I'll address that a little bit at the end. And so all of our data shows that RML GP61 vaccinated mice are just, their T-cell response is better than ATCC. And so the next thing that we wanted to do was to, well, I thought actually would be sort of a home run experiment. It would be very simple, straightforward. I was going to vaccinate mice, wait 28 days, challenge them with GP61 expressing SHU4, look at antigen-specific cells by flow, and slam dunk, paper, no problem, because RML vaccinated mice were just going to be better than ATCC. All of our data up until this point had shown that. The data that in the very first manuscript from Amanda Griffin, RML vaccinated mice do better than ATCC. All of my functional data for the T cells, RML is better than ATCC. But again, this is why you do the experiment, because sometimes things don't turn out quite the way you think. So again, we're following these cells, these antigen-specific cells, in mice that have been vaccinated with RML GP61 in filled circles and ATCC GP61 in open circles. And we've challenged them on day 0 with virulent SHU4 expressing GP61. And again, my y-axis here is counting the total number of antigen-specific CD4 positive T cells that have an effector phenotype. And I also want to note that day 0, that is day 28 post-vaccination. And so this is what was on board the time these mice were challenged. And so I think what will jump out to all of you is that ATCC GP61 vaccinated mice look fine. They're, in some cases actually, they have more antigen-specific CD4 positive T cells, for example, here in the lung. And then in the other organs, they look exactly the same as an RML GP61 vaccinated mouse. And this really baffled me, because I was actually expecting that I wouldn't even have any ATCC GP61 vaccinated mice left at day 14 to even analyze data from. And so this led us to a few questions. Well, maybe the SHU4 GP61 is intrinsically attenuated. Maybe something about overexpressing this epitope with this plasmid has somehow made the bug a little bit less virulent, and now the ATCC mice can survive. And we didn't really think that this was the case because it seemed to grow fine in bone marrow-derived macrophages. There were no indications at all that the expression of this epitope was having any effect intrinsically on the bug. And so our next hypothesis was that high-avidity CD4 positive T cells that recognize and respond to GP61 were contributing to protection. And what I mean by this is it's known in the literature that the T cells that recognize and respond to GP61 are extremely sensitive to this antigen. And so they can recognize this antigen when it's at very low concentrations. And so what we hypothesized was that there's actually a pool of cells, even in naive animals, that can respond so well to this antigen that that's going to have some effect on what we're reading out, especially in vaccinated mice. And so to test this, we got a little bit cute and used some facts of immunology to our advantage. So B6 and BALB/c mice have different MHC class II alleles. And so as I've indicated here with this yes and no, B6 mice can recognize the GP61 epitope, whereas BALB/c mice do not. And so this allows us to separate whether or not there is a contribution of a T cell response to the strain that may be the reason why all of the ATCC GP61 mice are surviving. And so we did this experiment in naive animals, again, naive, no vaccination, And what you can see is that when mice were challenged with GP61, the B6 mice, when they're challenged with RML GP61, they're significantly improved survival. And even though this is only 1 day extension in mean time to death, Francisella, it's extremely virulent. So you can see my naive mice die in about 4 days. The fact that when I walked into the mouse room on day 4, all of the valve Cs needed to be euthanized, whereas all of these 6s were still running around their pans, looking perfectly content. And it wasn't until 24 hours later that they actually showed signs of illness and needed to be euthanized. And so what this told us is that the GP61 expressing strains are better controlled in mice with T cells that can recognize and respond to this epitope. Now, the other thing that we wanted to know was whether or not GP61 specific T cells alone were sufficient to mediate protection. And to do this experiment, we vaccinated mice with either LCMV, which of course would express the GP61 epitope. It's a naturally occurring epitope in this virus. As a control, we used Pachyndé virus. And we used Pachyndé virus as a control in addition to naive mice because Pachyndé virus, the antibody response can cross-protect against LCMV. And so we wanted to have another infection that just lacks the GP61 epitope to really nail home whether or not that epitope was important. And what I hope you can appreciate here, when we challenge mice with what were supposed to be lethal doses of ATCC GP61 and RML GP61, you can see it wasn't quite 100% lethal for RML GP61. There is no difference in the ability of mice that were vaccinated with the epitope, without the epitope, or not vaccinated at all to survive these challenge doses, suggesting that GP61 specific T cells alone aren't sufficient to mediate protection. And instead you need GP61 plus a polyclonal anti-Francisella response. So now we sort of get to our, what I think is the real home run experiment here, which is looking at whether or not GP61 improves vaccine-mediated protection. I think I've sort of alluded to this when I've been going through my talk. And so in this experiment, I'm now showing you a few more of our controls that we did. So we vaccinated mice with either ATCC expressing IGLC, which is dashed in gray, or ATCC GP61, it's solid dashed line, RML IGLC in solid gray, and then RML GP61 in solid black. And you can see when mice that are vaccinated with these different strains are challenged 28 days later with SHU4 IGLC, the ATCC vaccinated mice don't do particularly well. Although we do see some survival in the ATCC GP61 vaccinated group, they're still not surviving to the same degree that, say, the RML GP61 mice are surviving. So that recapitulates what we expected. However, when we challenge mice with SHU4 GP61, you can see that 100% of the mice survived this infection, even the mice that were vaccinated with strains that didn't express the epitope initially. And so why is this happening? So here's sort of the model of what we think is occurring. So after ATCC vaccination, you get this small pool of antigen-specific T cells that, frankly, might be hard to detect using our class 2 tetramer. In contrast to this, vaccination with RML GP61 results in a larger pool of antigen-specific T cells. And then when mice are challenged with these different strains of SHU4, you can see that when they're challenged with SHU4 IGLC, they're not seeing this epitope a second time. And so there's not this really nice pool of high avidity T cells to respond a second time. The mice end up dying of infection. However, when ATCC GP61 vaccinated mice are challenged with CHU4 GP61, those T cells proliferate, they expand, they are able to mediate protection, and so those mice now survive. And then in our RML GP61 scenario, you already have a larger pool of antigen-specific cells to start with. And so even though you don't recall that GP61 specific response during CHU4 IGLC challenge, if we sort of use GP61 as a surrogate for the ability of the vaccine to elicit antigen-specific cells, we already know that RML is better at that. And so it's likely that those cells are the ones that can then mediate protection. And of course, then with RML GP61, you expand this pool even further, and again, all of those mice survive. So this leads me now to the conclusions. So I hope that I've convinced you today that vaccine-mediated immunity against tularemia is associated with mice having a larger pool of polyfunctional antigen-specific CD4 positive T cells. And importantly, if we can include an epitope, in this case it's GP61, that can elicit these high avidity T cells, you can convert a poorly protected vaccine, which is ATCC, into one that's highly effective. In fact, all of the mice were able to survive virulent CHUS4. Further, and I think what's really important to note is this entire time we've been using this exogenous epitope to do all of these studies. And so GP61 doesn't occur in Francisella. The reason that we used it was because we needed a tool to track those cells. However, I think that it is possible that Francisella expresses an epitope that would have the same sort of effect. And so not only would it be very useful to identify that epitope in a mouse so that we can continue to use our mouse model, if we're going to develop a successful subunit vaccine that can be used in people, we're going to have to identify epitopes that can sort of have this same property. And so that's something that our laboratory is really interested in pursuing. So of course there's many remaining questions. I think that we've done a really good job of answering that first thing I talked about at the beginning, is identifying correlates of protection. But there's still a lot of work to be done in understanding improving longevity of protection, and also the ability of mice and men to survive higher doses. And so one question is whether or not GP61 can improve longevity and allow mice to survive higher doses. I'm also really interested in why protection wanes so quickly. And so what is happening to effector T cells after vaccination? So if you don't have longevity of protection, that suggests the T cells aren't sticking around. So why is that? Why don't these cells develop after vaccination with ATCC? And why does LVS vaccination sort of seem to fail to elicit central memory T cells? That's also something we're really interested in pursuing. And finally, we're interested in where these protective T cells reside. Are they in the lung? Are they in the airway? Are they in the tissue itself? Or do they reside in the periphery and then are recalled to the lung upon intranasal challenge? So those are all sort of things that we're pursuing in the Bozio lab. So of course I'd like to thank my mentor, Dr. Katie Bozio, for allowing me to work in her lab and provide such a great project for me to work on. And then of course I've shown you a lot of mouse work, a lot of big experiments in the BSL-3. This would not have been possible without our two fantastic technicians, Debbie Crane and Tara Worley, who are so valuable to me in helping me complete a lot of this animal work. And then of course all the other members of the Bozio lab for their support and thoughtful critiques of my science. The recombinant strains were provided by Brad Jones at the University of Iowa, and the LCMV was provided by Jeff Bellinger and Jennifer Erlob at the University of Arizona. So I'd like to thank you all so much for your time, and I'll be happy to take any of your questions.
Thank you, Dr. Lydia Roberts, for that informative presentation. It is time for Q&A. Now, if you have a question you'd like to ask Dr. Roberts, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click the send button. We will answer as many of your questions as we have time for. Now let's get started. Dr. Roberts, our first question is, now have you started to look at whether GP61 improves longevity of protection or protects against higher doses?
Yeah, we have started those experiments. So in those experiments, we're just using our RML strain because we felt as though let's take the vaccine that we already know works better than ATCC and take those studies further. And so I've gone out to 90 days post-vaccination, which I know doesn't seem like a very long time, but so far we have been unable to protect a mouse 90 days post-vaccination. And so in that sense, our mouse model closely recapitulates what we find in people, is that protection wanes very quickly. If we include the GP61 epitope during both vaccination and during challenge, we do see 100% of those animals survive. Importantly, if we remove the epitope from the challenge strain, so if we challenge with SHU4 without GP61, all of those mice succumb to infection. And so even though we have improved RML in the sense that GP61 can lead to longevity of protection, we still, I think, have a long way to go because it's completely dependent on the presence of that epitope. We have also looked at higher doses. I've gone up to 2,500 CFU, which again, it's not that high of a dose. However, I'm not able to protect any of the mice, even with GP61 RML, GP61 SHU4. They all succumb to virulent Francisella. And so I think while we've made some progress, I think there's still a lot of room for improvement. And hopefully identifying epitopes and using different vaccination strategies can further improve upon the LVS vaccine.
Dr. Roberts, how did you purify the lung T cells?
Yeah, so this is a big problem in our lab when I started, because we had been sorting the cells using just a regular ARIA sorting. It took forever, and we were having a lot of trouble getting the assays to work. And I think a lot of it had to do with the fact that T cells just took so long and were pretty abused from digesting them out of the lung and then going through the sorter, all the centrifuging. It was a lot. And so what we have done instead, which works great, is we digest the lungs into a single cell suspension with Libraze, and then use the Miltenyi CD4 microbeads. And this allows us to get a very, very pure population of CD4 positive T cells very quickly. Within an hour or so from the digested lungs, we can start to finish, be ready to set up our cold culture system, which is fantastic. And not only do my experiments work much better than they did before, but they also take a lot less time.
Would you expect to see a similar lack of immune memory with other bacteria strains?
If you're referring to, I'm trying to think. So in terms of Francisella, I don't think we would see it with a less virulent strain. I think that the lack of immune memory, it's hard to protect against SHU4. I think one problem is trying to protect against intracellular bacterial pathogens. I don't think that I'm trying to remember how many vaccines are on the market for intracellular bacterial pathogens, and it's very few. So I think in general it's sort of hard to protect against intracellular bacterial pathogens. probably because we're not eliciting a good enough T cell response. I think it's easier to elicit protective immunity when the pathogen is extracellular because antibodies are very effective at mediating that protection. So I think there might be something unique about eliciting protective T cell responses that is different than eliciting protective B cell responses.
We have some great questions coming in, but we're running out of time. This will have to be our last one. Dr. Roberts, can you elaborate on what you know about the CD8 response?
Oh yeah, great question. We, in addition to generating the GP61 strains, we have also generated a strain expressing the immunodominant CD8 epitope from LCMV GP33, and we've also attempted to make the OT1 expressing strains, so synfecal, because these are both really well characterized responses. We haven't done a lot of work so far with the CD8s, but that is sort of a story that's really getting interesting, I think. Interestingly, we do see the same phenomena where if we elicit high avidity CD8 T cells, we can convert ATCC from a non-protective to a protective vaccine. And the other interesting component of the CD8 T cell response that I'll mention is that CD8 T cells can control Francisella infection. They're not as effective at doing so compared to CD4 positive T cells, and that might just be a function of the cells. It also could be that the bacteria is very good at manipulating the CD8 positive T cell response, particularly since it's an intracellular pathogen. You think CD8s would be really good at killing those infected host cells via cytotoxic activity. But I'm guessing that the bug is trying to do everything in its power to stop that, to maintain its replicative niche. And so I think there's a lot of interesting things going on with the CD8s that we're just starting to delve into.
I would like to once again thank Dr. Lydia Roberts for her presentation. Dr. Roberts, do you have any final comments for our audience?
I'd just like to thank everyone again for listening to my webinar today. And again, thank you to Miltony and to Labroots. It's been a really fun morning for me to share some of my work with you all.
Thank you again, Dr. Roberts. And thank you to Miltony Biotech for making today's educational webinar possible. Now, before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 15th, 2017. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. Now, that's all for now. We thank you for joining us and hope to see you again soon. Goodbye.